All right, welcome. Today, we're diving right into the seriously cool, high-stakes world of radiopharmaceutical chemistry. We're going to look at how scientists perform what is basically a kind of molecular magic, taking totally normal molecules and making them radioactive so they can hunt down and fight disease. So, this is the big question, right? It's a puzzle, but one with incredibly high stakes. Think about it. Chemists have to design a molecule that can navigate the super complex environment of the human body, find one very specific target, say a single type of cancer cell, and then carry a radioactive payload. And that payload either has to send out a clear signal for a scan or deliver a killer therapeutic dose, all without falling apart on the journey. It's a huge challenge. You know, at its heart, a modern radiopharmaceutical is just a modular marvel. It's built in three parts. First, you've got the targeting ligand. This is your heat-seeking missile, programmed to find a specific receptor on a cancer cell. Then you've got the radioisotope. That's the warhead, either for imaging or for therapy. And connecting them, that's the crucial linker. Or if you're dealing with radioactive metals, this special molecule called a bifunctional chelating agent, which holds onto that radioisotope in a super stable grip. So, to really get how these things are built, we're going to walk through the three main chemical strategies chemists have up their sleeves. We'll look at an electrophilic swap out, then the absolute workhorse of the field, nucleophilic substitution, and finally, a really cool technique that uses a chemical claw. And then, we'll see why all this delicate chemistry is almost always done by high-precision robots. Okay, let's dive into our first method. Honestly, the best way to picture electrophilic substitution is to think about swapping one specific Lego brick for another on an already built model. You're just making one tiny, precise change without messing up the rest of the structure. So the chemistry here is actually beautiful in its simplicity. You take a radioisotope that's an electrophile. That just means it's electron deficient. It's hungry for electrons and actively looking for a place to react. So chemists designed a precursor molecule with a spot that's really rich in electrons, like an activated aromatic ring. This makes it an absolutely irresistible target for our radioisotope to swoop in and just kick out whatever was there before. And this method is super popular for attaching things called radiohalogens. For instance, you can stick radioiodine right onto tyrosine bits in peptides. But an even cleverer trick is something called iota demetallation. What chemists will do is pre-install a metal-containing group, like one based on tin, right onto the molecule. This group basically acts as the perfect placeholder, which the radioactive iodine or astatine can then swap out with incredible precision. It's a really neat trick. All right, let's move on to our second method. This one is, well, you could argue it's the most important one out there, especially for modern PET imaging. If our last method was about an electron-hungry atom attacking, this next one completely flips the script. So here's the contrast, and it's really interesting. We're now using a radioisotope that is a nucleophile. It's electron-rich, and it's looking to donate its electrons. So the chemist's job totally changes. Instead of making the target molecule attractive, now they have to equip it with a really good leaving group, basically a chemical fragment that is more than happy to get pushed out and replaced by our incoming radioactive atom. And this method, especially with fluorine-18, is the undisputed king of clinical PET imaging. I mean, the production of FDG, that's the glucose analog used in literally millions of cancer scans all over the world, it relies on this exact chemistry. That negatively charged fluorine-18 ion comes in as the nucleophile, shoves a leaving group out of the way on a sugar molecule, and boom, you have your final radiopharmaceutical.
Okay, but what if your radioisotope isn't a nice halogen like fluorine or iodine? What if it's a metal like gallium-68 or technetium-99m? You can't just swap it into a carbon-based molecule. It just doesn't work that way. You need a completely different strategy. You need a way to grab onto that metal ion and hold on for dear life.
And this is where chelators come in. The name is perfect. It literally comes from the Greek word for claw. A chelator is an organic molecule that has multiple binding points, and it uses them to wrap around and securely cage a metallic radioisotope. This creates this incredibly stable little package, which is then attached with a linker to the targeting part of the drug. This chart shows it brilliantly.
Chemists have this whole toolbox of chemical claws, and each one is designed for a specific metal. For technetium-99m, which is the workhorse of SPECT imaging, they might use a chelator like DTPA for kidney studies. For gallium-68 PET scans, these claws called DOTA and NOTA are the gold standard. They're famously used in agents that target neuroendocrine tumors. The whole key is just matching the right claw to the right metal, so you can be sure it doesn't break free once it's in the body.
So now that we get the how of the chemistry, we have to talk about the who, or maybe the what, that actually does the work. Because in a modern radiopharmacy, it's almost never a person standing there with a beaker. It's a robot. This quote really just nails it. You know, handling highly radioactive materials, especially the ones that decay really fast, is incredibly complex and hazardous. To get the kind of safety, precision, and consistency you need for a medicine that's going to be injected into a person, well, automation isn't just a nice to have. It is an absolute must. And everything, I mean everything, comes down to this number: 109.8 minutes. That's the half-life of fluorine-18. That means in less than 2 hours, half of your radioactivity is just gone. The entire process, making it, purifying it, doing quality control, it all has to happen on this incredibly tight schedule. There is zero time for slow manual work. The clock is always ticking. Loud. When you look at the problems, it's so clear why doing this by hand is just a non-starter for making these drugs routinely. First and foremost, the radiation dose to the chemist would be dangerously high. Second, even the best chemist in the world can make a tiny mistake, which could mean a whole batch fails quality control. That's a hugely expensive error when your main ingredient is literally vanishing every second. And finally, it's just too slow. And this is why automation is the perfect solution. It solves every single one of those problems. It boosts safety by putting thick lead shielding between the person and the radioactivity. It gives you incredible reproducibility, hitting the exact same temperatures, pressures, and timings every single time. It's fast, and it guarantees you're complying with what's called good manufacturing practices, or GMP, often by using these sterile, single-use cassettes that make cross-contamination impossible. So this is what a modern radiopharmacy lab actually looks like. The synthesis isn't happening on a benchtop. It's happening inside these things called hot cells, these heavily shielded boxes with thick lead and leaded glass. Inside, there's a synthesis module, which is basically a compact, computer-controlled robot, and it just executes the entire chemical sequence flawlessly, from the first reaction all the way to the final purification, getting the dose ready for the patient. So, at the end of the day, it's these elegant chemical strategies, supercharged by the speed and safety of automation, that make modern nuclear medicine possible. They let us create these exquisitely sensitive molecular probes that literally give us a window into the biology of disease. And that just leaves us with this one tantalizing question. Now that we have the tools to build these molecular spies with such amazing precision, what new disease target will we teach them to find next?